Light blue. Additive, sodium citrate. Tests performed, PT/INR, prothrombin time/international normalized ratio, PTT, partial thromboplastin time, APTT, activated partial thromboplastin time, fibrinogen, D-dimer, heavy metal testing, clotting disorders. Function. Sodium citrate is an anticoagulant that works by binding calcium ions in the blood, which is necessary for clotting. This action is reversible, allowing technicians to perform clotting time tests on blood collected in these tubes. Proper fill requirement. It is important to fill the tube to the correct level to ensure the proper ratio of blood to additives, which is critical for accurate test results. Under or overfilling can affect the results of coagulation tests. Use. These tubes are specifically used for coagulation studies and are essential for testing clotting disorders or assessing the coagulation profile of a patient. The reversible nature of sodium citrate allows for the examination of clotting times without permanently affecting the blood sample. Red. Additive, none, no anticoagulant or gel separator is present. Purpose, designed for serum collection. Blood collected in these tubes naturally clots, allowing the isolation of serum from the cellular components after centrifugation. Tests performed. PSA, prostate-specific antigen commonly used for prostate health evaluation in men. Therapeutic drug monitoring, TDM used to measure drug levels in the blood to ensure the proper dosage of prescribed medications. Serum lead testing, measures lead levels in the serum, often for toxicity assessments. Serology testing, the scientific study of blood serum, particularly concerning the immune system's response to antigens, e.g., detection of antibodies for infections like hepatitis, syphilis, and HIV. Key notes. Red top tubes are ideal when a clotting process is required to separate serum for various biochemical and immunological tests. After collection, the blood must sit for 15 to 30 minutes to clot before centrifugation. These tubes are primarily used in serology, toxicology, and drug monitoring. Gold, serum separator tubes, SST. Also known as SST, serum separator tube, gold tube, or tiger tube, red-gray marble. Additive. Contains a gel that separates serum from the cellular components after centrifugation. The tube also contains a clot activator to promote rapid clotting of the blood. Purpose. Primarily used for the collection and separation of serum for diagnostic testing. The gel acts as a barrier during centrifugation, preventing contamination of the serum by cellular components. Tests performed. Infectious disease testing, HIV, hepatitis panels, e.g., hepatitis B, C, RPR, rapid plasma reagent for syphilis, HPV, human papillomavirus. Hormonal testing, pregnancy, beta-HCG, TSH, thyroid-stimulating hormone, FSH, follicle-stimulating hormone. Metabolic testing, cholesterol, HDL, LDL, total cholesterol. Paternity testing, genetic testing for parentage. Antibody and DNA testing, useful for HLA typing, human leukocyte antigen, and other antibody-based tests. Key notes. This tube is widely used in immunology, serology, and chemistry panels for the detection of diseases, hormone levels, and metabolic markers. It is not suitable for blood cultures or microbiology testing, which require sterile conditions. Alternative tube. In some cases, a red-gray marbled tube, tiger tube, may also serve the same purpose as a gold top tube. Special instructions. Allow the blood to clot completely before centrifugation, typically 30 minutes. Always ensure proper spinning to separate the serum and maintain the integrity of the sample. Light green. Plasma separator tubes, PST. Additive. Sodium heparin or lithium heparin, anticoagulant, to prevent blood clotting. Often includes a plasma separator gel that creates a barrier between plasma and blood cells during centrifugation. Purpose. Designed for plasma-based chemistry tests for faster processing since the blood does not need to clot before centrifugation. Tests performed. Chemistry levels, Basic Metabolic Panel, BMP, and Comprehensive Metabolic Panel, CMP. Liver panels, ALT, AST, bilirubin, and total protein. Electrolytes panel, sodium, potassium, calcium, magnesium, and chloride. Lipid panel, total cholesterol, HDL, LDL, and triglycerides. Albumin, a protein test for liver and kidney function. Amylase, to assess pancreatic function. Arterial blood gas, ABG, evaluates pH, oxygen, and carbon dioxide levels. Key notes. Unlike serum separator tubes, SST, light green tubes are used for plasma, not serum. These tubes are ideal for time-sensitive chemistry tests where immediate plasma separation is required. Do not use light green tubes for therapeutic drug monitoring because heparin may interfere with results. Special instructions. Properly mix the tube after collection to ensure the heparin is evenly distributed and prevents clotting. Ensure the tube is filled to maintain the proper blood-to-additive ratio for accurate test results. Lavender. Additive. EDTA, ethylenediamine tetracic acid, an anticoagulant that works by binding calcium ions to prevent blood clotting. Purpose. Primarily used for hematology and some specialized chemistry tests. The EDTA preserves the shape and structure of cellular components for accurate analysis. Tests performed. Complete blood count, CBC, includes multiple tests such as black small square WBCs, white blood cell count, evaluates immune system activity. Black small square RBCs, red blood cell count, assesses oxygen transport capability. Black small square platelets, evaluates clotting function. Black small square H+H, hemoglobin and hematocrit, checks for anemia and hydration levels. Hemoglobin A1C, hemoglobin A1C used for diabetes monitoring, providing an average blood sugar level over the past 2 to 3 months. ESR, erythrocyte sedimentation rate, also called the Westergren sedimentation rate, measures inflammation in the body. Sickle cell screening, identifies sickle cell anemia or trait. Ammonia levels, monitors metabolic disorders and liver function. Note, samples for ammonia testing should be kept on ice and processed immediately. PCR, polymerase chain reaction for DNA or RNA-based testing, often used in infectious disease and genetic analysis. Key notes. Sample type. These tubes are used to collect whole blood or plasma, depending on the test requirements. EDTA preserves blood morphology, making it ideal for hematology tests. Not suitable for tests requiring serum, as the additive prevents clotting. Proper filling and mixing are crucial to prevent clotting and ensure test accuracy. Special instructions. Gently invert the tube 8 to 10 times after collection to mix blood with the anticoagulant. Some tests, such as ammonia levels, require immediate processing or refrigeration. Gray. Additive. Potassium oxalate, an anticoagulant that prevents blood clotting by binding calcium. Sodium fluoride, a glycolysis inhibitor that preserves glucose by preventing the breakdown of sugar in the blood sample. Purpose. These tubes are specifically designed for glucose and lactate measurements and alcohol, ethanol, testing. Sodium fluoride ensures accurate glucose readings by inhibiting enzymatic activity. Tests performed. Fasting blood sugar, FBS, measures blood glucose levels after fasting. Glucose tolerance test, GTT, used to diagnose diabetes and gestational diabetes. Lactic acid measurement, monitors lactic acidosis, often in critically ill patients. Samples for this test must be collected without a tourniquet, as it can artificially elevate lactic acid levels. Ethanol, ETOH, testing, determines blood alcohol levels. Key notes. Sample type, plasma is typically used for these tests. This tube is not suitable for general chemistry panels because the additives interfere with other tests. Accurate glucose measurements are ensured for up to 24 to 48 hours after collection due to the glycolysis inhibiting properties of sodium fluoride. Special instructions. For glucose or lactic acid measurements, samples must often be placed on ice and processed quickly to ensure accuracy. Properly mix the tube by inverting it 8 to 10 times to distribute the additives evenly.